So, what are clones? Clones are organisms that have exactly the same genetic material, so their DNA is exactly the same. Clones can occur naturally, for example, identical twins are clones of each other, but cloning can also be done artificially. But how is that done? The most basic type of artificial cloning is to clone a plant by taking a cutting. If a branch from the parent plant is cut off, and the leaves are removed and the stem is planted in moist soil, after a few weeks the stem will take root and grow. However, cloning animals is not quite so simple. Animal cloning needs to be done in a lab for one thing. There are two methods of creating clones of animals. The first is artificial embryo twinning. Identical twins naturally occur when a recently fertilised egg, for a reason that nobody completely understands, divides into two separate embryos. They then start dividing by themselves to become separate individuals. The individuals, because they came from the same fertilised egg, therefore having the same genetic material, are completely genetically identical. Artificial embryo twinning uses the same process, however it is carried out in a petri dish, which contains agar jelly and nutrient broth. The cells are artificially divided and then they are implanted into a surrogate mother, where the cells can finish developing into individuals. The other method for cloning animals is called SCNT. This stands for somatic cell nuclear transfer. So, what is a somatic cell? A somatic cell is a term used for any body cell which is not a reproductive cell, i.e. a sperm or an egg cell. Reproductive cells are sometimes also referred to as germ cells. In a mammal, a somatic cell has two sets of chromosomes, whereas a germ cell only has one. Now, before we get ahead of ourselves, let us clarify what chromosomes actually are. Chromosomes are found in the nucleus of most cells. They are made up of DNA. A gene is the part of DNA that has the code for making a particular protein. Each chromosome contains several thousand genes. When the two germ cells fuse together, each set of chromosomes combine to create an embryo which now contains two sets of chromosomes. However, in a somatic cell nuclear transfer, the somatic cell provides both sets of chromosomes. So it starts like this. The egg of a donor organism has its nucleus removed, so all of its genetic information has been taken away. Next, the nucleus of a somatic cell is then isolated and transferred into the egg cell. They are then fused together with an electric current. The cell starts developing into an embryo, and then it is implanted into a surrogate mother and continues to grow. The individual is genetically identical to the donor of the somatic cell. So what other types of cloning are there? When cloning complete organisms, it is known to be reproductive cloning. Cloning doesn't just have to be about entire organisms, you can also clone genes and cells. Cloning a gene involves isolating just one gene and copying a strand of DNA into a plasmid. A plasmid is a small molecule of DNA, usually found in bacteria, that is separate from the majority of bacterial DNA found in the nucleoid. By inserting the gene into a plasmid, it is much easier to manipulate the DNA. As it is a bacterium, the bacteria will replicate, also creating identical copies of itself and the DNA sequence. Therapeutic cloning uses the procedure of SCNT to clone cells. After fusing the somatic cell nucleus and the egg cell, the embryo is then encouraged to divide into a cluster of cells called a blastocyst. The blastocyst contains an inner layer that contains many stem cells. These stem cells can become specialized and give rise to many different cell types, including neural, blood, and cardiac cells. So what are the uses of cloning? Stem cells that are harvested can be cultured into organs that people can use to transplant. As the organ is made from the same genetic material, the risk of rejection is significantly less. Reproductive cloning could be used to help endangered species, as they could reproduce more and they wouldn't have to fear extinction. More trees and plants could be grown faster in order to solve the deforestation problem. It would also provide a habitat for the endangered species. Cloning more plants and livestock could help solve the problem of malnutrition in third world countries, providing starving communities with food. There are some disadvantages though. Some people believe that it is unethical to interfere with nature and play God by creating life. Also, the infused egg is often unstable and takes hundreds of attempts before a successful embryo is obtained. In 2006, a sheep called Dolly was cloned using SCNT. She was the only successful egg out of 277 attempts. And despite these drawbacks, cloning has much to achieve for humanity. In the future, it may lead us to do things from curing life-threatening diseases to saving species on the brink of extinction. The possibilities for good are endless.
Created using Powtoon.